Can you guys hear me?
Yes, Senji, you were talking about species richness and abundance when you got disconnected.
Yes, I'll do that. I'm really sorry. I'm in a place where there's very poor network right now. My Wi-Fi went off and the power went off. I'm really sorry. I'll just in case you guys miss something, it should be there in the recording because I'm doing an offline recording anyway, so that should be there in the video. So I'll post both the videos in case if there's a disconnection. So I'll just quickly try sharing my screen again. Yeah, could someone just confirm if it's visible?
Yes, it's visible.
Uh, okay, all right. Yes, so I was mentioning about species richness and abundance. So this is the one that people had questions on. So if those people are on the call and still on the call, if this is still not clear, you can stop me and ask me about clarifications for the same. So the species abundance and richness basically is what determines what kind of microbial communities are present in that environment. So an individual microbe forms a microbial population and they form a guild. Microbial guilds are usually different kinds of microbes that cohabit a region or coexist in a region. They come together to cohabit in a particular environment. Uh, yeah. So the diversity of the communities indicate the quality of the environment. So how depending on how diverse the environment is, how diverse the biological, the microbial communities that we see, we can determine how good or disturbed the environment is. Species diversity or richness depends on the environmental conditions, because the resources available in a region will what is support the diversity of organisms. Many different conditions like the micronutrients and macronutrients, temperature, water availability, pH, oxygen levels, light, every basic biotic and abiotic factor will affect the species diversity. So this is how the different species exist. But what is the role that they play in an environment? So what actually determines the role of a species in an ecosystem? So for that we need to understand about this concept called niche. Each species, not just microbes, every species occupies a niche in the community. This niche is what is the different kinds of interaction it has, the role it plays in the environment where it lives, what it eats, how it reproduces. Everything can be understood from the concept called niche. So this is an example of different species of warblers coexisting in a particular niche. So what is the difference between an habitat and a niche? Habitat also is a similar concept, right, where an organism exists. So an habitat, sorry, there
I
is there any question? Excuse me. Okay, if there's any
nothing,
question,
ma'am.
yeah, yeah, is there any issue? Are you guys able to hear me?
Yes, you are audible.
Yeah. Okay. So I'm assuming that my connectivity is fine and I'm audible. So if there's some issue, please let me know. Yes. So a parrot, a honeybee, and a squirrel, all of them can exist in a similar kind of an habitat, in basically the habitat of a tree. But what exactly do they do in the tree? Where do they live? What kind of interaction do they have? Is what are the different niches of habitats on a tree? So parrots lay eggs, they build their nest on a tree, they use it for reproduction of their species. Honeybees on the contrary uses the flowers for collecting food, so for protection of food they use this. And honeybees also build nests on trees, on buildings, so that can also be classified under niche. But preliminarily they collect food from a particular region in a tree. And squirrels help in dispersal of seeds. They eat these seeds, and then they help in dispersal of these seeds and help in growth of the seed itself. So these are the different roles that they play in that particular niche. It could either be for their reproduction, it could be for foraging, it could be some other role that helps the tree itself. So these are the different niches that are occupied by these individual organisms. Different species can basically coexist in one habitat by sharing different niches. And for instance, this whole tree could be a niche for any of these organisms. So that's exactly like the fundamental niche that is available for all of the organisms. But these organisms divide it among themselves. They use these different regions of the tree for different purposes, and that is the realized niche. So some of them inhabit only the upper region, some of them inhabit the middle regions, and some of them inhabit the lower region. So these are called the different realized niches. So this is about the concept of niche and their interaction with the environment. But they also interact with various abiotic factors. Okay, this dog wants to attend the class along with me. Please, no, Prasad. Okay. I'm really sorry. By next class, I'll be back in ISC, back with better network and without any of this uh, natural environment interactions happening during the class. So yeah, so these are the different interactions that a microbe can have, or any organism in general can have. So biosphere, atmosphere, lithosphere. So lithosphere is the soil, hydrosphere is the water bodies, biosphere is plants, animals, everything that is biological organism, the flora and fauna. And atmosphere is all the galaxies and heavenly bodies and anything that's in vapor and gaseous phases. uh, Types of biogeochemical cycles. So this interaction primarily of the microbes with the environment mainly happens through the biogeochemical cycles. The hydrological cycle or the water cycle, through which the water gets recycled and starts pouring as rain, just like how it's just pouring down here right now. And atmospheric cycle is the carbon cycle, oxygen cycle, and nitrogen cycle. Sedimentary cycle includes phosphorus and sulfur cycle. And there are also other many other types of cycles, including rocks, because rocks get weathered and broken down into the soil, the mercury cycle, and so on. So these are the different classifications of biogeochemical cycles. Biogeochemical, this word itself encompasses different components. Bio indicates the biological components that interact as a part of the cycle. Geo indicates soil or the lithosphere chemistry. And chemicals, these individually, each of these are different chemical components that play a major role in this cycle. So, please. I'm sorry, guys. So the nitrogen cycle, this is what I was talking about earlier when I was talking about denitrifying bacteria and plants that take the help of this bacteria for utilizing nitrogens. So anything death that is dead is decomposed by decomposers, and those nitrates go back into the soil and they are fixed by nitrogen fixating bacteria for plants to utilize them, and that's how the nitrogen gets cycled. And plants, while fixing while utilizing the Nitrogen again will give back them during the cycle, and this is how it gets cycled through the atmosphere. So here there are different players in this, the biological organisms, the microbes, along with these different chemical molecules constitute the atmospheric biogeochemical cycle. So yeah, this is the last slide I have for today's session. Uh, Microbes interact with each other. They can be interacting positively or negatively. Species richness and abundance depend on the environmental conditions, and various environmental conditions can actually determine the richness and abundance. Niche defines the specific roles. So the abundance and richness are just about their existence, but what do they actually do is determined by the term called niche. And microbes interact with abiotic factors and play a major role in biogeochemical cycle. And understanding microbial ecology and biogeochemistry can help us in exploiting microbial properties for environmental problems, because they form such a very important part of the biogeochemical cycles, and if something happens to them, that is when these cycles get disrupted. So understanding what exactly the microbes are doing in these different environments can help us in tweaking them for fixing some of these environmental problems that we face. So that is all I have in the slide deck for today, and there's still 10 more minutes remaining. And yeah, quickly, if you guys have some questions or suggestions, please do post them. And yeah, how denitrifying bacteria present in the soil gets attracted towards leguminous plant roots? Is there any energy which attracts the bacteria towards the root? No. So these bacteria are always there in the soil. It's just that leguminous plants are better at supporting their growth. So whatever the leguminous plants produce, the kind of food they produce, or in very simple terms, let's just say the kind of food they produce is good enough to support these denitrifying bacteria to grow. So bacteria just don't grow on all the substrates. They need a specific kind of a substrate and specific kind of an environment for them to grow. And leguminous plants generally support the growth of these bacteria. So the bacteria are always there in the form of spores, or in a vegetative state, always in the soil. They just don't grow, but they start growing when they find a suitable condition, and that suitable condition is provided by the leguminous plant roots basically. I hope that's clear, Sumit. And I think you had also asked some other questions on email, or was it someone else? So if I think there was someone, Kavita, I think. If you're still on call, and if you're in this call, you can ask if those doubts are still there and if it's not clarified. Any more questions? Any more suggestions, doubts? Anything about the lectures, or do you want something specific for me to uh, prepare for the next class? Like I said, I'll be back in ISC next class, so I can make a better undisturbed version of the presentation.
Ma'am,
Yeah,
ma'am, I want to ask one thing that is not related to today's topic, ma'am. Outside of it,
yeah.
ma'am, ma'am, the exam of this NPTEL course, ma'am, will it take place through offline mode, ma'am?
Yes, yeah, you have to register for the course, and it will be in offline mode for the certificate. That's what I think. But you can get more information about this from the NPTEL team itself. So if you mail to that thread, uh, where on the discussion session or the discussion forum, you will get more information about the exams from the NPTEL team.
Okay, ma'am. Thank you.
Yeah,
Thank you.
thank you.
Hello,
Any?
ma'am.
Yes,
Uh,
yeah. Hi, Sumit.
yeah, मैम एक्चुअली ये था कि जैसे एक पर्टिकुलर माइक्रोब की स्पीशीज है, उसको अपन आइडेंटिफाई कर रहे हैं, उसकी जेनेटिकली मॉडिफाई करके उसको एक पर्टिकुलर सिचुएशन के लिए अपन ने मॉडिफाई कर दिया। ओके, बट मैम मेरा क्वेश्चन ये है कि एक्चुअली जो उसकी आसपास सराउंडिंग जो कम्युनिटी, जो रिसोर्सेज उसके पास जो प्रेजेंट है,
हाँ.
तो मैम उस वो सारे फैक्टर्स को अपन कैसे अपने फेवर्स में यूज कर सकते हैं, क्योंकि उसका क्या प्रोसीजर वगैरह होता है, क्योंकि इट्स एनवायरनमेंट के अंदर उसके देर आर थाउजेंड ऑफ पॉसिबिलिटीज एंड कॉम्बिनेशंस, जो
Yes.
कुछ भी आउटबर्स होते हैं, तो उसको एक कंट्रोल एनवायरनमेंट में वो कैसे अपन उसको हैंडल करते हैं वो।
Okay. So you're saying that we are genetically modifying. Okay, I can respond in English, right? Because my Hindi is not that great for responding. You can understand what I'm saying, right?
नो मैम, यस मैम।
Yeah, okay, thank you. Yeah, so if I got your question right, you're asking that we are taking a microbe from a complex environment and how can we employ that or exploit that in a controlled laboratory conditions. Is that what your question is?
Yes, ma'am. Other factors, resources which are affecting those microbes.
Yes, yeah, yeah, you're absolutely right that when a microbe is present in a complex environment, it will have other interaction with other microbes, other organisms, and other abiotic factors as well. That is definitely true. But what happens in a laboratory condition is we try to mimic some of the conditions that's not completely out there, but these interactions are we just have to see whether those interactions are necessary for their growth and development itself, or whether those are secondary interactions. So what kind of conditions that we give will have like a substrate and the necessary nutrients that is just enough for it to grow and perform the function that we need. So if it is a function like chewing up the oil spills, then that particular gene is modified in such a way that irrespective of what environment it is in, it will just take that substrate and keep growing and not interact, and the other interactions that it generally has are not needed. So the genetic modifications are done in such a way that those other interactions anymore play no role in their level development or life cycle, and they the conditions are just enough for them to grow and do the specific purpose. So we can't completely bring in the natural environment under laboratory conditions, but it will be in such a way that it is basic enough for them to have the enough nutrients to grow and perform the function of interest. Was that clear enough?
Yes, ma'am. But मैम ये कि 100% श्योरिटी नहीं होती, इट्स जस्ट लाइक कि जितना क्लोजली हो सकता है उतना
Sorry.
श्योर। It's just like that. Okay, we can't be 100% sure. It's just that like how close we can just be.
Yes,
That's
yeah, it's just
just
trial
that.
and error. So not all microbes found in the environment can be modified. Even genetically modification cannot be done for any microbe that is out there. So that is why we specifically select. So for instance, E. coli is a bacteria that has been used genetically for various purposes and various applications because of the ways that we can manipulate it. So not all microbes can be modified, so we select them in such a way that they work best under laboratory conditions.
Yes. Yeah. Thank you, ma'am.
Yeah, thank you. Any more questions, suggestions, clarifications? Yeah, we still have like two more minutes remaining, I suppose. So if you guys still have any questions, you can always mail it to me. I'll definitely respond back to mails. uh, and I can, if you can comment on the YouTube as well, so when I get a better network situation, I'll upload the videos to YouTube. You can always check those videos and comment if you have any questions or comments. Is that all? Nice presentation, thank you so much. And I'm really sorry about the mess that happened in between, but yeah, thanks for the compliment. Thank you so much.
Thank you, ma'am.
Thank you. Thanks, everyone.
Thank you.